welcome to the 22nd episode of Breakfast at UMHealth webinar. For this second session, we have with us Dr. Safwani, Kamarul Zaman from Faculty of Pharmacy. So Dr. Safwani is a trained pharmacist and now is a senior lecturer at the Department of Pharmaceutical Life Sciences of the faculty. She currently serves as the president of Tissue Engineering and Regenerative Society of Malaysia, TESMA. Her research is mainly focused on stem cell technology, with special interest in the application of biosensors and artificial intelligence. And today, Dr. Safwani will share with us her research updates entitled Oxygen Matters, the Effects of Physoxia on Mesenchymal Stem Cell. So please welcome Dr. Safwani.
Very much, Dr. Amira, for that kind introduction. Good morning, everyone. I'm Dr. Wan. So today I'm going to present to you some updates from our research group, which is the Oxygen Matters, the Effects of Physoxia on Mesenchymal Stem Cells. Okay, so let's start off with some definitions. Sorry about that. Okay, so these are the three terms that we are normally familiar with. I'm sure you probably have heard of these terms before. So in cell cultures, we have the term normoxia, hypoxia, and recently the term physoxia. So I think we are familiar with normoxia, which is the oxygen level of about 20 to 21%. Okay, so it's just a rough estimation. I think it's a good approximation, okay, currently that we use in cell culture, okay, in general. And there is also hypoxia, which is the pathological hypoxia, normally defined as oxygen level below 2%. Although this is not absolute, some literature actually mentioned that the level of hypoxia is between 2 to 6%. Okay, and there's a physoxia as well. Physoxia, meaning physiological hypoxia, normally defined as the oxygen level in which the tissue within our body respond to maintain their normal physiological condition. So the range is about 2 to 6%. All right, so there's a bit of confusion in terms of what the literature or publication, okay, in terms of cell culture, because the term hypoxia has been used widely in terms of defining the oxygen level or the low oxygen level that has been used in cell culture. So I think when I talk to some of the researchers in the field, they mentioned that the term physoxia may be more suitable to be used to define the oxygen level, okay, the normal oxygen level within the body, okay, that is more suited, okay, for cell culture. All right. Right. Now, according to some literature, there are certain oxygenation level within our tissues. Okay. So I think there are various oxygen level that we should take note of, okay, especially when we are culturing cells, okay, in terms of stem cells. If you're thinking about differentiating the cells, perhaps oxygen levels should be taken into account because those will mimic, okay, the normal physiological condition in the body. So for example, like adipose tissue, previously I worked quite a lot with adipose tissue. Is actually quite highly vascularized, but in terms of its native microenvironment, it is only about in human is about less than 4% oxygen. Okay, so that gives us some idea on why oxygen level should be taken into account, okay, when we are culturing cells, especially stem cells. Right, so why do we need to address this issue? So current practice normally, I think those who are doing cell culture in the lab would agree with me that normally our cells, okay, actually maintain under 21% oxygen, which is unlikely the optimal culture condition. Okay, and this obviously, as the reason which I already mentioned before, it does not mimic normal physiological condition. So the oxygen level, okay, the alteration in terms of oxygen will definitely affect some of the stem cell parameters, okay, that we have in the lab currently. So basically, stem cell, particularly stem cell, because I work with stem cells, they are currently they are cultured at oxygen level that is not suitable, which not mimicking, okay, the physiological condition as what is inside the body. So should we consider Physoxia for stem cell culture or stem cell expansion? So some of the literature already mentioned that in terms of metabolic activity of stem cells with regards to the expression of HIF1 alpha gene at 2 to 6% oxygen, the stemness activity, if you look at your left, okay, the stemness activity is either enhanced or maintained. So which means that the stem cell are able to maintain the stemness, okay, which will increase their possibility of differentiating into certain cell lineage. All right, and then there's also decrease in terms of differentiation or spontaneous differentiation activity, which is most likely due to the notch signaling pathway. And there is also decrease in terms of oxidative stress in the cells, as well as the increase in terms or decrease in terms of the metabolic activity, specific metabolic activity, increase in self-renewal proliferation activity, which I'm going to show to you later on, some of the findings that we have from our lab. And of course, there's an increase in terms of the motility as well as the adhesion of stem cells due to the increase in growth factor release, particularly VEGF. Okay, so I'm going to show you some of the findings that we have gotten, okay, from our lab work. This is from the human mesenchymal stem cell derived from adipose tissue. Okay, so some of the findings that we have seen before this is that when we cultured cells at 2% oxygen as compared to 21% oxygen, the stem cells are actually increased in number. So if you can see from the images on the left, when they are cultured at 2% oxygen, at day 3, there's not much of difference. But if you look at day 7, okay, all the way up to day 10, you can see that there's increase in terms of the proliferation rate of the cells. Okay, and they're also able to maintain their fibroblastic feature, which is the main feature of stem cells. All right, so as you can see from the graph here, is that you can see that the cell number increase, okay, by day 10 as compared to normoxia, okay, the 21% oxygen, and this is actually significant from day 7. All right, and the population doubling time is also decreased, which shows that the proliferation rate is very much enhanced, okay, in hypoxia. So cell viability, when we do cell cycle analysis, we have observed that the cells in the cell cycle phase, okay, most of the cells are in the S phase. All right, so we can see that the cells are higher in this phase. And when we look further, when we do the annexin 5 assay to look into apoptotic cell number, so we can see that in Physoxia at 2% oxygen, that the cell viability are actually higher with less number of apoptotic cells and necrotic cells. And when we do COMET assay, okay, to determine the percentage or the rate of DNA damage exerted by Physoxia by 2% oxygen, we can see that the cells are actually the DNA damage is actually less, as seen from the tail length, the percentage of tail formation as well as the tail moment. So if you can see from the images here, maybe it's not that clear. We can see that the formation of the tail or comet, okay, of the cells are actually less in hypoxia. So that the formation of tail actually indicate DNA damage. So in this case, under 2% oxygen, there is actually less DNA damage. So when you look at the gene expression, okay, using RT-PCR, So at 2% oxygen, we can see that the HIF1 alpha expression is actually increased as compared to the normoxic condition. And this is coincide with the increase in the growth factor, which includes the VEGF, BFGF, HGF, and IGF. All right, so not so much on HGF and IGF, okay, but it's more significant in terms of the secretions of VEGF and BFGF. So we think that VEGF and BFGF could be the one that drives the increase or the enhancement of the proliferation rate of the cells. And this has been validated by by ELISA. In terms of stemness marker, so these are the markers that we use in accordance to the minimal criteria set by the ISCT, okay, the International Society for Cellular Therapy, which showed that the CD markers of the stem cells are actually not much difference between normoxia and physoxia or hypoxia in this case, okay, at 2% oxygen. So there's not much of difference there. Okay, so we can say that in terms of CD markers, the stemness is pretty much maintained in both conditions. However, when we look into specific gene expression, which defines stemness, which is the REX1, SOX2, OCT4, and NANOC, we can see that in 2% oxygen, okay, in physoxia, that these genes are actually increased as compared to when they are in normoxia, so which also tell us that the stemness activity within the cells are actually enhanced or maintained. In terms of differentiation ability, so the main criteria to define stem cells, one of them is to look into their differentiation ability into adipogenic, osteogenic, as well as chondrogenic. So in normoxia and hypoxia, we can see that they are both positive for all three differentiation types, okay? But when it comes to the adipogenic differentiation, we notice that it is actually less in hypoxia. This is also true for osteogenic differentiation as compared to normoxia, but under hypoxia, chondrogenic differentiation is actually increased, okay, as when you're compared to normoxia, okay, by looking at the protoglycan formation as stained by blue, detected by Gaussian blue staining. Now, when we look at the differentiation gene expression level with regards to all differentiation types, so this is it's parallel as to what we have seen with the staining images, with the staining assay, that we can see that the adipogenic differentiation in terms of the adipogenic genes, these are the three main genes, okay, that signifies adipogenic differentiation is actually significantly lower. If you look at the white bar, okay, between normoxia and hypoxia, they are actually lower in hypoxia, okay? So same goes for osteogenic, okay? For all these three genes, the main ones that signifies osteogenic differentiation, so we can see that the genes are actually expressed lower when they are in normoxia. But on the other hand, chondrogenic under hypoxia seems to increase, okay? If you look at all the three genes, okay, the main three genes, the SOX9 and ACAN, as well as the collagen 2A, okay, the expression seems to increase under hypoxia, under 2% oxygen. Right, so I think the next question that people might ask is that whether physoxia or the low oxygen level pose risk in terms of the tumorigenic potential. But as you can see from this gene expression of these tumor suppressor genes, the PRP, P16, P21, and P53, it seems to show that there is a significant low expression of these genes under 2% oxygen. And there's also no significant changes when we test it, okay? When we look into the telomerase and the telomere activity, there's not much of significant, there's no significant difference between the telomere activity as well as the telomerase activity in both normoxia and hypoxia, which probably indicates that the Physoxia, or at 2% oxygen, there is no increased risk of developing tumorigenic activity. However, this would require further investigation. So this is something that we are trying to find out in the lab as well, whether Physoxia is really safe condition to be used when we are proliferating the stem cells in the lab. Now we go a little bit further to look into whether Physoxia can be used to culture the cells without using any serum. So, as we can, I think, as we all know, that serum, particularly the fetal bovine serum, has been used widely in the lab. So some of the initiative that has been done even now is to look into alternatives that would require less dependence on serum. Okay, so some of these initiatives is to look into the possibility of using low oxygen concentration to help with the proliferating of the cells without having to use serum. So in our lab, what we have seen is that without serum, okay, when we culture the cell under 2% oxygen, the 2% oxygen seems to increase the number of cells, okay, especially at day 7, as you can see from here. But of course, when you use serum, the proliferation activity is very much higher, okay, even though they are under hypoxia or under 2% oxygen, okay. But when we take out the serum, okay, it seems to support the proliferation activity still, even under 2% oxygen, okay. But as compared to normoxia, the 21% oxygen, and the cells does not seem to, the increase in proliferation is not as high as the one in 2% oxygen. So this could probably indicates that even without serum, Physoxia condition may be able to support the proliferation activity of the cells. So when we look at the viability and proliferation rate, okay, so it seems to be parallel with the images which I've shown you before this. So we can see that the cells which is cultured without serum at 2% oxygen has a higher viability as compared to those that is cultured without serum in 21% oxygen. Okay, so when we can also see that the cells, when they are cultured in with serum at 2% oxygen, seems to have the highest number of cells, okay, as compared to other conditions. All right, so we can see that at day 7, okay, the cells that is cultured without serum, okay, show a significant increase in the cell number when compared with without serum in 21% oxygen. Right, so our findings indicate that Physoxia at 2% oxygen seems to support, okay, proliferation rate of the stem cells in both serum and serum-free condition, although with serum, okay, the proliferation rate is very much enhanced, all right? And normoxia at 21% oxygen is still better in terms of supporting different differentiation types, okay, especially into adipogenic and osteogenic cells. Whereas Physoxia at 2% oxygen seems to support better differentiation into chondrogenic cells. And Physoxia seems to downregulate tumor suppressor genes, although we think that this require further investigation, all right? So because one of the things that is of concern is that the tumorigenic activity, especially now that the industry, stem cell industry, are actually looking into ways on how to increase or enhance cell number, okay, in terms of their cell culture method and protocol, which is something that is very much needed. So right now, one of the ways they are looking into is how to utilize hypoxia or Physoxia, okay, to enhance the proliferation activity of the cells. So the implication on the therapeutic application of stem cells, we can say that certain application may work best under Physoxia and hypoxia, as indicated from our data just now, okay? And monitoring oxygen levels is very critical, okay, especially when when you want to differentiate the stem cells into a certain cell lineage, so oxygen levels has to be taken into account. And of course, specific oxygen level may be important to be determined, okay, for certain cell differentiation or application. So that is something that has to be determined first before we proceed with our application. Now, of course, there's always the safety concern. So I mentioned just now that we have looked into the risks of whether Physoxia is able to increase any tumorigenic potential or activity. But with regards to the expression of HIF-1 alpha, which is the key factor in regulating oxygen with regards to hypoxia, is that depending on the condition that they are in, okay, or where they are expressed, HIF-1 alpha does have the ability to support tumorigenesis. All right, so of course, the HIF-1 alpha can be, I think most of us would also know that HIF-1, especially HIF genes, okay, have been found to be activated in tumor cells. Because they are also able to promote key steps in tumorigenesis, that includes angiogenesis, metabolism, proliferation, metastasis, as well as differentiation. And on the other hand, the HIF-1 alpha also have anti-tumorigenic activity, whereby it can indirectly induce cell cycle arrest by inhibiting the MYC activity. So in this regard, it can also induce apoptosis. Okay, this could be due by its activity by inducing the expression of BNP13 and the NIX genes, which promotes pro-apoptotic activity. So again, it's pretty much, I would say, it's a double-edged sword condition, whereby this actually tells us that precise control of the environment, the stem cell niche, is very important. So in this case, in terms of controlling the oxygen level, we have to ensure that the HIF-1 alpha activation is able to benefit the stem cell therapy, so that the whatever therapy that we offer to the patients will be safe and efficient. So this image is just to show you with regards to the mechanism, okay, that's involved in tumorigenic activity, is that the hypoxia, through the expression of the HIF-1 alpha protein, is able to promote, okay, the mechanism of angiogenesis, erythropoiesis, metabolism, as well as the cell proliferation. Okay, so those are the main concerns when it comes to Physoxia or hypoxia at the low oxygen level in cell culture. So in summary, I would like to emphasize again that the physiological oxygen condition in cell culture can actually provide us, may be able to provide us with better understanding of physiological as well as the pathological processes of stem cells, so which emphasize the need to understand more in terms of the biology behind the oxygen condition. And of course, this would also allow us to have a more reliable and relevant experiments, especially with regards to the effects of different stimuli in stem cells, especially in stem cells. But this is also can be applied with other types of stem cells as well, other types of cells as well. All right. I think that's all from me. Thank you very much. Dr. Amira, back to you.
Thank you so much, Dr. Safwani, for that insightful sharing, especially to reinforce like how important oxygen tension is in like microenvironmental cue to ensure that we can grow this cell more effectively. So just a quick question. So I'm really, like I said, it's just a paradoxical role of HIF-1 alpha, right? So do you think that mesenchymal stem cell can differentiate into cancer-associated fibroblasts if we were to grow them at a very kind of Physoxia environment? Is there any?
Yes.
Is there any possibility?
That's what we are concerned. Yes, there is a possibility of it becoming cancerous or tumorigenic, in cell culture. That's why I think one of the things that we have to develop in terms of the protocol is what sort of assay that will be good to test, to analyze the cell before we use it for other applications, or when we push it out to our patients. So those are the things that we need to develop actually in the lab before we can even think about applying it to the patients. Yes.
Yeah, it's just so ironic because like you said, HIF1 is such a tumorigenic precursor, you know. Okay,
Yep.
so maybe we can move forward to our next question. So I think it's from Sarah Ahmad. So she's saying that a lowering percentage of oxygen to 2% is a good condition for stem cell experiment. But how difficult is it to be able to do this in the lab? Like, how do you actually conduct this experiment?
Right. Okay. So earlier on, we conducted our lab. We do have an incubator that is equipped with oxygen sensors. So we are able to control the oxygen to the level that is required. So in this case, we are able to tune the condition inside the incubator to 2% oxygen. Now, having said that, I know that this question might come up again, is that how do we ensure that, I think one of the things that people have been asking is that how do we ensure that the 2% oxygen, although the incubator is set at 2%, whether that 2% oxygen actually reaches the cellular level of the cells. Okay, so it is actually quite difficult. It is not easy to actually mimic that condition. So what we are trying to do right now is to develop a sensor that we can actually attach to the bottom of the flask to detect the oxygen level. So I think there is a commercially available ones, but I'm not sure, haven't tested it yet. How accurate is the sensor in terms of detecting 2% oxygen? So yes, we have oxygen. We do have an incubator which can support 2% oxygen, but the 2% oxygen in the incubator may not be the same as the one at the cellular level. So what we are talking about is, could be that the level of oxygen that reaches the cellular level could be a lot less than what we have, as compared to what we have set in the lab, in the incubator. So those are one of the difficulties that we are facing right now, is how to really mimic properly. Right now is how to get the oxygen level at the 2% oxygen level to reach the cellular level, to reach right at the cells. So at the cells, what we are hoping for is that hopefully what we are getting at the cells, at the cellular level, is really 2% oxygen. But right now, I still have my doubts that what we are getting at the cellular level is not really 2%. So that is actually the difficulties that we are facing right now. So we are trying to mimic as much as we can to the physiological condition. At this moment, we think that 2% oxygen is still the best in terms of proliferating stem cells in the lab. Yes.
So are you having doubts that the oxygen reaching the cells are lower?
Could be. So we are still testing it actually. So as far as incubator is concerned, according to the sensors that we have set lah, basically, right? So we assume that what we are getting is 2% oxygen.
I see. Okay, I think that concludes for our session for today. Once again, thank you very much, Dr. Safwani, for your sharing, and thank you everyone for your kind attention. Thank you.